So we get started, I do want to give a little intro of our guest speaker today. So our guest speaker here is Liana, and then she's also a alumni from our 2024 summer camp. So really glad to have her today. She will be sharing her journey and her experience in STEM and sharing a research project that she conducted. So we are really excited to welcome her today, and we hope everyone can respect her time and also be respectful during her presentation. So now I'll be passing off to Liana and then I'll be sharing my screen.
Hi everyone. Hi, my name is Leanna Chen, and then I'm a past alumni from GSM program, and I was in GSM last year. I really enjoy this program, so I hope to contribute back by also being a guest speaker and sharing my experiences in my research. And yeah, I have a great slideshow um here for you guys, and then so what I'm doing is research on Drosophila melanogaster. Maybe a lot of you guys don't know what that is, but that's basically like the fruit flies that you guys see, but it's a special breed of fruit fly that we use for research. And LO 1548 is the line number of our research, and it's conducted by my team Van Gogh. Yeah, next slide. Yeah, a little about me. I currently attend Lowell High School, which is a school in San Francisco, California. And my field of interest is basically, as you see, it's developmental biology and biotechnology. And I hope to major in that field in my college as well. I have experiences in the UCLA Emerging Scientist program, and I'm a coordinator of that program. I also do fly research, neuropharmaceutical research, and I also publish an editorial about STEM equality and equity. So I believe like many of you have seen like fruit flies whenever it's like, near um fruit areas or like fruit produce a secret like smell, and fruit flies are attracted to that. And these fruit fly are a little bit different from the ones you see in the wild or at your home. It's a special type of fruit fly that we use in our fly research. And basically these flies and their genetics are artificially modified. And basically the genotype is written as below. They have four chromosomes, but we only listed three because the last one is not as important. So for this fruit line, we made its genetic genome and like its phenotype to have red eyes, curly wing, mutant humerus, tubby body, and ebony colors. And if any of you guys know how to read like genotypes on the third chromosome, it is basically the artificial thing that we put into the fruit fly. So that's called a double balancer, and this will balance our gene and keep its genotype stable while we keep breeding the flies across many of the other generations. And then you might be wondering what our research is about. And our research is about um, putting in the um, artificial P element into the fruit fly. And our research work basically is trying to find like where the the artificial P element that we inserted was at. The P element is a artificial jumping gene. It's a transposal element. So basically, you jump randomly in the fly's genome, and basically, what we're trying to find is where it jumped. And through like multiple crosses, we actually did this over a year, and we found that the diagram is basically where we proposed the insertion site, and we think that the P element jumped on the chromosome 2L. As I said, there was four chromosomes in total, and it jumped on the second chromosome on the left one. And then we think the P element insertion would be 3 to 5 orientation, but it can also be the other way around, which is 5 to 3. And we concluded the orientation ways by the diagram like to the left. And you can see that that's like the exact insertion site that we found, which is 2L15062, and then yeah, to all of those bases. And we think that the five region is untranslated, which means we conducted research, but we weren't able to translate that part. And the direct repeats that we found is the triple T cat cat G. That's how you found this in our IPCR data for the SPEC1P LAC4. And we also did Query 1 found in the Flybase, and Flybase is basically a website for the fruit fly, specifically to research about their genes and all that. And then we were trying to find the 3' end, but we didn't have any conclusion for the 3' end. And the primer design for the 3' end is needed for our next step of research, which I hope to conduct for like the next year or so using Dove1. And the diagram was basically talking about the primer picking results that we did. And I'm not going to go too deep in it because, yeah, it requires way more explanations and context to fully see that graph. Next. And this is something called JBrowse. And this is basically kind of like a visual like diagram that we see where our gene jumps into. And the yellow region is where it jumped into. And there is two genes. So like the two blue ones, it says VAS to solo. And you can see how this is 5 to 3' as I labeled the 5' on that side. And then the gene it jumped into is VASA and solo. And some gene information about VASA is that it's like a protein. This like gene sequence encodes for a protein that is like APT dependent protein, and it plays like really important roles in like cellular like processes and like development very heavy. And you can find like most of these functions in like germline cells and reproductive cells in fruit flies, and you see it most active during the developmental phase. So we concluded that this sequence would be most important for development in Drosophila flies. And VASA also have like jobs in supporting the development in male Drosophila flies. And we also think that without this, then the Drosophila flies wouldn't be able to, conduct its reproductive task. And this image on the left, it's literally the most accurate protein picture that we can find because it's X-rayed, and this is basically not like predicted. It is exactly how it shows and exactly what it's like. And we found this image on the UniPro site, and UniPro is a very helpful like website where all of you guys can have free access to search up like what gene codes for what protein. And this green color of this protein doesn't mean that the protein is actually green. It just means that it's the most accurate version of protein diagram you can find. And then the other gene that we inserted and jumped on was Solo, sister on the loose. And this encodes like a chromosomal gene that involves like meiosis and like all that separating cells. I think if you guys were like in biology classes, you guys might have like hear of like separating like chromatids, sister chromatids, all of that. And basically it is in charge of that. And it's also kind of involved in the nucleus division in meiosis for female flies. So earlier, VASA was more important in male flies, and this one is more important in female flies. And this have lots less of research compared to VASA, which is where we want to conduct more research on. We want to find out more about Solo because there's already a lot of research that have already been done on VASA. So we see this as a breaking point. And you might notice on the left, the image is not green no more. And the reason because of that is that this is a less researched gene. So all of these structures are kind of like what do you say, like predicted depending on its base pairs and its chemical properties. And this is not like X-rayed, so like this is the most like accurate form that we can predict it to be like, which is different from VASA where we like X-rayed it and confirm that's how it's shaped. And then these are the predicted expression that we think these two genes have. And for VASA, you can see that VASA like express more in the embryo phase. And that's related to what it's for, right? For the development and in germline cells. And it's more expressed in the male too, if you want to keep that in mind. Compared to the female germline cells, you can see that the male germline cell box is redder and darker than the female germline cells. And then for solo, it's less expressed, but you can also see that it have red boxes in the female and the male germline boxes. So in conclusion, our P element jumped on the genes that are heavy in developmental. And without these proteins, we would assume that developmental phase and the embryo phase for these Drosophila melanogasters would not function. They would not develop into adults because if missing of these two genes, developmental and reproductiveness of these slides would be difficult. And then we also use this very fancy microscope. It's called a Sora microscope. And we went to UCLA to borrow this microscope because it was way too expensive. It was like millions of dollars put into this microscope that we just can't afford in our lab. And we imaged the L3 larva brain. It's basically level 3 developmental phase of like a kind of like a fly baby is what we call larva. And we dissected the brain. It took a lot of work. It took weeks to get like a perfect brain. And we dissected and then we brought it to UCSF to image it. And basically we put some blue dye that you can see in the image. And these are called DAPI dyes. And the green is basically the GFP expression. And under this camera, you can see like exactly like where it glows. And you might notice that like the green is not that obvious. And it's because as I said, this gene is for developmental purposes. So it has less expression into the brain. And so it have less like what do you say, green and like GFP expression. If you compare it to like flies that my colleagues did, they were really heavily green because their genes have related like expressions in the nervous system. So that's where it have more GFP expressions. And then human ortholog and paralog is, you know, evolution purposes, we have similar genes to Drosophila. And that's why we kind of researched this gene because a lot of their genomes and sequences are related to humans. So we can learn a lot from their genes and they're like a great model organism. And for human orthologs, we see one in like Visa Hepsa DDX4. And basically, this is a dead box protein. And it also plays roles in development, embryos, sperm production, and all of that in our bodies as well. And we also see a paralog, and it have like a less like similar score of 9 out of 13 compared to 10 out of 14. And this is also very important for embryo, but not for development, but for fertility and like viability, and also maintaining that the male germline stem cells. And then this is just like the diagram and like how the proteins look like for the human ortholog. And we can see that this is showing the domain of like helicase ATP binding. And for both the human orthologs, and also the human ortholog is different if you didn't know, for Vasa and Solo, and they look different as well. And the more green it is, typically means that it's more accurate as in what we're predicted. And then next up is the pair log, and these images are we found it on UniPro, and it can also show you the how like helicase ATP binding looks like, and also helicase C terminal. And these images are very wonderful because it takes long times to predict them and for scientists to look for them. And then, as I said, our research is not like perfect and finished, and we hope to confirm the three prime end with like primer design. And we didn't find anything for our research that we conducted this time, but we were thinking of doing sequence analyze, and then we design a new primer for the three prime end to find what's on the three prime end. And also, we want to redo IPCR for more data on the three prime end, so our research will be more accurate. And as you guys see for the brain dissection and imaging, we ran into like some problems and we weren't able to like dissect the most like perfect brain ever. And our image was in like very like blurry form. So we hope to like do that again and see if we can get a more accurate image of our brain. And then this is acknowledgement and citations to the Stanford lab, to like Dr. Coco and Dr. Park. And we also got help from the UCSF Parnassus campus to like let us image and gave us a lot of like advice and help. And we also like to thank our Lowell PTSA, which is a grant that basically funded us and helped us through all of this research. Thank you so much, guys. And I'll be open for questions. You can feel free to like unmute yourself or type it in the chat.
So let's put it in the chat actually. And Lily can pick the questions.
Yes. First of all, I want to thank you, Liana, for this amazing presentation. It's definitely a lot of like a lot of research and really informative. So I really appreciate that and for sharing your research project. So for anyone who have questions, you can just put it in the chat and then I'll be coordinating the questions. So you guys can go ahead and put your questions in the chat.